Ladies and gentlemen and ladies, cats and dogs, children of all ages, and everything in between. Welcome, welcome, welcome. You know who it is. It is the kid, Unc Just, the pizza freak, resonating at the pizza frequency. What is that? It means I love talking about pizza. I love making pizza, but mostly I love sharing pizza with people that I give a shit about. Yes, literally breaking bread with people that I care about. And I have a sneaking suspicion that you too, my friends, also resonate. It said pizza frequency, or why else would you be here? And where is here? Well, I am so glad that you ask. Here we are, Science of Sourdough, Part 2, with Cameron Martino and James Gaffney coming through. Noel, this is our Part 2 of Science of Sourdough. Noel and I are doing Slow Rise is doing office hours every other Monday, and we're starting to do some more specialized classes or free open webinars, trying to increase awareness and just, answer everybody's questions. We want to be here because this is we're passionate about pizza people, and we have a sneaking suspicion that you are too. So very excited. So here's the deal today. Cameron is going to be talking to Noel the whole time. We're going to do a PowerPoint presentation. So I would encourage everyone that if you are on your cellular phone, turn it to horizontal view so you can actually see these PowerPoint controls or see the slides that we're going to be sharing with everybody. So very excited. Next week, Monday night at the same bat time, we'll be doing office hours, but the following week, I believe that will be part 3 of the Science of Sourdough. And I believe we'll be with Stephanie Swane from Modernist. So really excited. Stephanie's a really good friend of ours. We've had a chance to hang out with her a number of different times. So always really excited to hear what she has to say and her insight and all that good stuff. So without any further to do, I'm going to bring on the man, the myth, the legend, Noel himself. So thanks everybody. Enjoy the webinar. Noel, if you're there, come on. Blue bell. There he is. There he is. Now I got to spotlight you. I'm going to spotlight for everybody. And then I'm going to spotlight me real quick. Here we go. Add spotlight. How are you doing, Noel?
Better now that we're both spotlighted. Thank you, Justin.
Yeah, that was a little bit. We're getting this pin spotlight thing down. We're locking it down, everybody. I promise. One of these days we'll get it. This is the first time Noel and I have ever been on Zoom.
Feels that way sometimes, right?
Well, every week, every time I log in, it's a new update and this and that. Anyway, how are you doing? What's going on? What's the dealio with this evening, huh?
It's all good. I just came from Orange County doing a consult with a 35-year-old pizzeria that's looking for an upgrade. So I'll be keeping an eye on my Instagram, people, if you want to know what's going on in the OC, Huntington Beach specifically. So that was fun. It was a little bit muggy today. It actually rained in the morning. So for people who say that humidity doesn't affect their dough, I beg to differ.
Yeah, it was sticky. It's been sticky. I got to make some dough this week, and I know that I'm going to be doing the dance a little bit. Hey, real quick, next Monday office hours. Then the following Monday, can you just put a plug in for what that sourdough is going to be?
Yeah, so we're working with Modernist pizza, Modernist cuisine, Modernist bread. There's a woman whose name we always mispronounce. I'm going to call her Stephanie Swan because
Swane. I feel bad.
Swan. Okay. Well, we did that with Ezzo pepperoni.
That's right. That's right.
Ezzo, I called it Ezzo.
Hey, right? Look, my bad.
Swan. Let's call her Swan. She deserves the respect. So she's the publisher, and she's also extremely knowledgeable. So, Cameron and I have been trying to put together awesome presentations. So 2 weeks ago, we had John Porter from Smog City Brewing Company here in Torrance, and we really did a deep dive into what bakers can learn from brewers. And it turns out we've been connected at the hip for centuries, really.
Yeah.
So there's a lot that I have already learned from John Porter, and I know that I learned a lot just from listening to him talk, and also I really appreciated the interchange between Cameron, who did a brief cameo 2 weeks ago.
Yeah,
he just did a little kind of a 10-minute plug for microbiology. So this is his return to the spotlight, and we're going to do a longer presentation,
more in depth. Yeah, more in depth.
More in depth. Yeah, not just longer.
Right. So, all right, I'm going to take myself off. Everybody who is out there, all of the attendees, I will promote you to panelists. We ask that you please have your microphones off. And if you do show up, we're gonna remove you until it's question time. So, thank you very much, everybody. Noel, take it away.
Here we come. So, hello, everybody. Welcome. Super excited to be here today. We've been practicing this one because there's a lot to it. There's a lot of science, and I noticed last week or 2 weeks ago when I was watching the webinar, and Justin had edited it, and we were putting it up on YouTube, that it wouldn't have been great if we had some slides. So Cameron and I made sure that we have some beautiful slides, and there's a lot of great graphics out there. So thank you, Cameron, in advance for all the hard work that you and James did to put this week's presentation together. So I will bring on Cameron momentarily. I just want to let people know that this is a free pizza webinar. It usually lasts about an hour, and we'll do our best to keep it to about an hour. Sometimes between the office hours and the science of sourdough, we might go a little bit over. So we started at 5 p.m. I'll bring Cameron on in a minute after Justin rings the big blue bell of pizzaness, and we can hopefully keep it to around 1 hour. But if you need to leave, you will have opportunities to ask questions in the chat. We actually appreciate if you allow us to make you a panelist. We appreciate that you show your faces, and I want to start a conversation with you if you have a question. And so this is not your normal webinar where you're an attendee and you can just type questions into the Q&A and we'll read them aloud. If that's how you want to roll, that's fine with us. But if you want to chit chat with us, we'd love to chit chat with you too. So it's going to be a relatively quickly moving webinar. We've got our presentation in PowerPoint, and I printed it out in black and white, old school I am, just so I can kind of take notes and keep things moving. One of the things about what's happening today is that some of these slides could really be 1 hour webinars themselves. And so this is one of those projects where we're just kind of, I don't want to say we're making it up as we go along, but I'm hoping that this is going to last many weeks and months. So far, people have been emailing me and messaging me through Instagram with lots of interesting questions. We've tried to even incorporate some of those questions from a baker in New Hampshire today. We just picked a few of them that we thought were particularly interesting, but it's part of a much larger conversation. And so we're happy that you're all here. We're happy that there's so much love and interest for the science of sourdough, because one of the things that really gets me going is how much misinformation there is out there. And you know what? I'm probably guilty of it too. When I was learning about sourdough, I thought that if you had an older sourdough, maybe it was better, or if your sourdough was from some exotic place like Italy, that it was better, or things like that. And so I think that if you want to include religion and mysticism and all those fun things in your sourdough culture, that is awesome. And there's no science that says that it doesn't actually count, right? Because if we talk to Cameron, we're going to find out that there's a lot of unknowns out there, which is awesome. And what I love about Cameron is he says I don't know to about 50% of the questions that I ask him, because he's a real scientist, and so he's not going to be making anything up. So here's the thing: I don't want to put any more words in Cameron's mouth. So Justin, why don't we ring that big blue bell? See if we can get Cameron the man on.
Let's do it, Cam. If you're there, come on down. Let's see. Cam, if you don't mind turning on your microphone, please.
Hello.
Loud and clear.
This is fantastic. All right, I'm going to jump off. You guys enjoy.
Beautiful. Cameron Martino, how are you?
I'm doing well. How about you?
I'm doing well too. Thank you. Sweating it out in Santa Monica, but I'm loving every minute of it. Thank you for joining us once again.
Of course.
Where are you today, sir?
In San Diego. Muggy and hot here as well. Good for the microbes and bad for me.
My dough likes 55 to 60, and I do too, turns out.
Yeah, good for microbes and good for dough.
There you go. All right, so I'm super excited today. So where shall we begin for people who are uninitiated? What are we doing here?
We're going to talk some about the microbes in your sourdough and a little bit of basic microbiology. We're going to hit it from 10,000 feet. and kind of stand on the shoulder of giants, as they were, of other scientists and their work, and try and give everybody a nice high-level overview. And so if there's something particular in here that we talk about that you really like and would like to delve into, because any of these slides could be an hour long, we could get one of those people to come on and talk about it in depth. I'm sure they'd be happy to, share their science.
Cool.
But
I can't wait.
Let me share my screen and I will get started.
Good. So guys, let me just a quick reminder, if you're looking at this on your iPhone, you might want to turn your phone horizontally. It'll help you see the images that we're going to project somewhat larger. So in some of the cases, there are some links that we'll provide in the chat or in the Q&A for you if you want to do a little bit of research on your own. Some of these will be in URL form, and others will be kind of citations, and you can just kind of copy and paste them into Google and see where they land. Unfortunately, as Cameron was telling us, some of them are also behind paywalls because apparently a lot of this science is either very expensive or very exclusive. I'm not sure which, maybe both sometimes. So Cameron, without further ado, what are we looking at here?
We're looking at some sourdough microbes in a scanning electron microscope image is what it's called, but what it lets you see is a single cell. And here we're looking at a few of them. The link to this is down in the bottom right. I did not generate this image, but we've got some bacteria and some yeast. The bacteria, I believe, are in green and yellow. And we've got some yeast in blue and purple there. They colored them for us so that we can see the difference in the cells, because if it's black and white, it's really hard to tell. It's a beautiful image.
It is a beautiful image. There is some art to science, isn't there?
Indeed. All right, let me also bring up my laser pointer so we can point to things. All right, so what is a microbe? So microbes are anything that you can't see with your naked eye. So if you need a microscope to see it, it's a microbe. And the first microbes were seen by Antony von Leeuwenhoek a long time ago in the 1800s, and he used this very expensive, fancy-looking microscope here, which is just a pinhole microscope. So you just look through that pinhole there, and he drew some of the things he saw because no one had ever seen these things before. And you can see his original drawings here. They're kind of digitized, made a little sharper, but you can see he drew a figure for each one. So he saw some kind of rod-shaped thing, and then he saw this little one moving around. So he kind of drew a dotted line to see how it was moving. And so we're kind of looking at some of the first microbiology in history here, which I think is pretty cool. But these microbes have been around for a long time, much longer than us, more than 3 billion years old, most of them. And they represent most of the life on Earth. So here we're looking, where I'm pointing, at the root of the phylogenetic tree of life. So this is the common ancestor of all life on Earth. And from there, it just diverged and diverged. And if there's a join, it just means that there's a shared ancestor there. Like a dog and a wolf have some kind of wolf-like shared ancestor that they split from. And each one of these tips here at the end of the tree is a different whole group of life. So there's a whole separate set of things within these that are collapsed down. And just to represent how diverse all of these microbes are, this, where I'm pointing here in the bottom right, is all the animals on the planet that you know and love are just in this one tip. So pretty much most of the moving and animal life that you see on Earth is just right here. And then you've got some plants and fungi here. And then all of this other stuff is microbes. So beneath what we can see is a huge world of diversity of life that keep us alive. They produce 70% of the oxygen we breathe. They made the oxygen that allowed us to live on the planet. And they continue to do so. And they're pretty amazing. And here we've got a little picture of a bacteria, which is one whole clade of life here. And then an archaea, which is a not as well-known set of life, but they're very broad. And those are what make, if you've ever seen the pictures of Yellowstone's lakes, the super colorful ones, that's a biofilm. So a thin film of archaea live there, and they're what create the color. They are the color there. And then we have a whole group of eukaryotes. So that's anything with more than 2 cells, multicellular life. And here we're looking at Aspergillus fungi, which is what makes a lot of the mold on your bread and all kinds of other things. You can ferment food with it too. And yeah, those are microbes. Noel, have a kept you.
I'm speechless. I love this slide. I don't understand more than 1% of it. Barely understand what you said, but I think it's an excellent jumping off point and a good summary. So thank you for that.
Of course. All right. So now you've got to bear with me through the second slide. This is the longest one, but hopefully it will be useful to everybody here. And we're going to explain what is going on in your sourdough at a very high level. So we've got a sourdough here. And if we buy a microscope, you can buy a microscope on Amazon for $100 if you want and get this kind of image. But if you look at under a microscope and you've got 100x magnification, the first thing you'll see are these big blobs. And what these are the plant cells. So there's dead plants inside your flour, right? They took whole plant, milled it down. And what that represents is food for your microbes. So it's a bunch of starch and sugars, which starch is just a giant chain of sugars, locked up sugars. And that's what's shown here, and I'm pointing at, is just a starch molecule where the sugars are aligned together. And then if you zoom in a little bit further, another 1000x magnification, so we're going deep now, you'll see the microbes. And what we've got here in the orange circle is a yeast. So you can see it's budding from itself. This is the original cell, and then it's creating a copy of itself here. And then in the green circle, we've got the bacteria, and they're doing the same. You can see there's little chains of them budding off of each other. And together, they're what ferment your dough in a sourdough. And they can be broken down into a subset of metabolisms. So what that means is they eat something, and then they expel it out, just like us. And what they expel is what gives you the leavening and flavor in your dough. So they're eating all of these sugars. And then depending on the sugar, it depends what they produce. So let's break it down a little bit. So we start with pentose sugars and hexose sugars, which are really similar. And you don't really need to fully know why they're different, but just bear with me that they're different. And what those do is they change the metabolism of the different yeasts, so what they eat and what they expel. And within the bacteria, there's a few groups. There's what we call homofermentative, which means they only produce one thing, homo meaning one. And then heterofermentative, which means they produce more than one thing. So if you look here, regardless of whether it's a pentose sugar or a hexose sugar, the homofermentative microbes will either produce 5 lactic acid or 2 lactic acid, and that's what makes the kind of dairy notes in your sourdough. So lactic acid is what like a yogurt might taste like. It's kind of creamy, a lot of diacetyl, which makes that creamy flavor, and then the lactic acid, which kind of makes that dairy flavor. Then we have the heterofermentative bacteria, and those take a pentose sugar and a hexose sugar, and they either make 1 lactic acid, 1 acetic acid, and CO2. So the yeast aren't the only thing leavening your dough. The CO2 is what is the gas that creates the bubbles. And so the bacteria make the bubbles too, and they help make the dough rise. And they can also make this lactic acid and 1 ethanol if you're feeding them hexose. And the acetic acid will kind of make that green apple kind of vinegary flavor to it. And the ethanol in low concentrations will kind of smell fruity and floral. In high concentrations, ethanol is alcohol. That's what, if you're a brewer, you're trying to get some more ethanol. If you're making bread, you want less ethanol. You don't want it to be super alcoholic. And then the yeast will make one thing with all of this, which is 1 ethanol and CO2. And they make the most of the CO2 that's going to contribute to the dough rise and provide the texture. And then all of these acids, which I colored in this gold and yellow, the lactic and acetic acids, will do 2 things. They contribute to the sourness and the overall flavor of your dough. And they will also exclude other organisms. So an acid decreases the pH. And what happens when the pH decreases, especially below 4.5 is usually the magic number for some reason, determined by nature, that it will exclude other organisms. So that's why your sourdough probably has a longer shelf life than when you make your bread with baker's yeast. That lower all of that acid excludes the aspergillus we showed a picture of earlier from making mold on your bread for at least a longer period of time. And then all of this contributes to the complex flavor and smell that you get from a sourdough. All right, Noel, have I lost you?
No, I'm here. Well, first of all,
that
you're
was
not
a
going
long one.
that was not even long. This is fascinating. You're not going to get me again, because when we did this in the dress rehearsal, I asked questions and you're like, well, no, that's going to be in slide number 4, of course. So I won't make those mistakes again. So here's something I'm just curious about. So I think we looked at some drawings, right, in one of the earlier slides from the 1800s. When did scientists first start to see like these visions in magnitudes of times 100 or even times 1000?
That's a great question. I don't know when that microscope was developed. That's a good question.
But is it within the last 75 years?
No, I would guess in the late 1800s, early 1900s is probably when they developed those types of microscopes. But what took us so long to start really understanding these communities of microbes, right? Because these microbes, like you see, there's different species, complete different kingdoms of microbes in here. We'll kind of explain what that means in a second. But to really understand these outside of just like, okay, this one's shaped like this and this one's shaped like that. And a lot of them that are different, perform totally different functions might be shaped exactly the same. Like you can't tell the difference between a homofermentative and a heterofermentative bacteria based off the shape. We needed sequencing technology, which is we explode the microbes, pull out their DNA, and then read that through a computer. And we won't cover that today, but that allowed us to kind of get a snapshot of what microbes are in a sourdough, what they might be doing, and all kinds of other environments. So in poop and oceans and all kinds of places. And that's how we found the whole kingdom of archaea. We didn't even know it existed.
That is fascinating. Let me ask you a question. When you talk about sequencing technology, what are you talking about?
So there are relatively recently, in let's say the last 10, 20 years, there's a lot of technology that allows us to take all of the DNA in a sample, right? And we're getting into the weeds here, but if you remember your hallmarks of molecular biology, maybe in high school, you had DNA to RNA and RNA to proteins. And that's what pretty much makes all life on Earth is you have an encoding, essentially a blueprint of what you want to make. And when you want to make it, you make all the bricks for that. And then there's another cell component which reads off pieces and tells you how many bricks you need. And then another piece that makes each brick and puts the bricks together. And that's what's happening. And what this technology allows us to do is take that information and read it into a computer format where we have a file with the A's, T's, G's, and C's that encode that blueprint. All of that information in a cell.
Got it.
Hopefully that was understandable.
So basically, it's the advent of computer technology that brought about the advanced sequencing, right?
That and some other technology in molecular biology, which we could spend the next hour and a half talking about.
No, that's enough. That's enough. So this is a fascinating slide, and we could probably do 3 or 4 different sessions just on this one slide. But for now, we'll leave it at that. Thank you for introducing some of these fascinating topics.
Yes, and hopefully this will all tie together in a practical way in a few slides of what does it mean and how can we shift our hetero and homofermentive microbes and how would that impact our final product, the practical part of practical microbiology. All right. So what's going on in your starter? I kind of hinted at this already. So we don't have units here, but we're just looking at the x-axis of these figures. So we have 3 figures. Each x-axis is a fermentation time from short, intermediate, and long. And the reason there's no units is because, of course, short, intermediate, and long, if you're a baker, you know that can change depending on what you're working with. how much you fed it, and all kinds of things. But what we're looking at is the microbial diversity in this first plot across the fermentation time. So if you, when you're starting out, when you have a short fermentation time and you kind of have a very young starter, you are going to have a relatively low fermentation activity, right? This is easy to understand, is that you're not seeing a lot of bubbles yet, not a lot of activity, and you have a low species diversity. What does that mean? It's the number of different species in your sourdough. So there's a whole set of species in your sourdough, and you can't detect most of them yet. They're at low abundance. They're just hanging out. They haven't started growing yet. And then as the fermentation goes along, you have this long tail where the fermentation kind of is not quite fully started yet, but it's starting to get there. And then it will start up really fast. You'll get this really strong fermentation activity for a period of time, and then it will peak, and then the fermentation activity will start to drop. And you probably have seen this before, where your sourdough starter will go, stop, and then start to shrink again. And what's happening is that the microbes in there have hit their carrying capacity, meaning there's too many cells, and they're telling each other, there's no more food, we have to stop multiplying. And then so they stop producing gas, and you'll see your fermentation activity drop off, and your sourdough starter will start to shrink again. And you're looking usually for an optimal sourdough in the middle there at a really active sourdough where you're going to pass that float test, where you drop the sourdough in the water and you'll see it float because it's got nice active bubbles in there. And during that time, your species diversity, so the number of different species detectable in your sourdough, are going to increase. At the same time, while all this is happening, your pH will drop. And this is coming from all those acids that are being produced. And as the pH drops, you're going to get a more sour flavor. What's also happening is the microbes that are left behind are going to be more acid tolerant. So not everything can live in a super acidic environment. That's why your sourdough has a longer shelf life. And so if you let your sourdough get acidic, you're going to leave only microbes that are acid tolerant, that can live in that high acidic environment. At the same time, if we're looking at the number of cells of yeast, and then lab just stands for lactic acid bacteria. So those are the microbes that we talked about that make the lactic acid. They can be homofermentive or heterofermentive. It's a mixture in that group. The cell density is the number of cells. You can see that matching the fermentation activity, it's kind of has this long tail, and then will start to exponentially grow for the yeast and then start to drop off. And the lactic acid bacteria will kind of just keep on chugging and then reach a stationary phase, is what we call it, which just means they've kind of reached their plateau of how many of them they can have in that environment. They're starting to run out of food, they're not multiplying, and they're just kind of staying steady. Questions, Noel? I get everybody?
I mean, I feel like we could have a webinar on each one of these. So there are no numbers, right, associated with the XY grid, right? So it's kind of relative.
This is just kind of a qualitative understanding of what's happening at a high level.
Right. So, but you couldn't ascribe any like you couldn't say like time and temperature would be what's driving these, right? That's not what
you would need a whole, it would be experiment dependent. So how much salt are you using? Are you using salt? What temperature is it? How much starter did you use? How many times did you pass your starter, right? So the units are left off purposefully here.
Got it. And that gray area again, what does that represent? That's like a peak.
That's kind of our mature optimal starter, ready to make dough with.
Beautiful. I love it. And then it looks like James pasted the figure 3 text into the chat there. Can you tell us what we're reading when we see this kind of scientific mumbo jumbo?
Let's see. So that's just the name of the paper. So if you want to look up the paper, you can search that text. Oh no. Hold on, I got stuck in the chat.
That's all right. So you could do a search for a review of sourdough starters, blah, 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 right? That's what you would search for?
Yes.
Right. And then
right,
that's awesome. So thank you for that.
Yes, we can. James could probably send the link too, in case it can make it easier for people to find it. All right.
And then I don't want to give away any surprises, but one of the women that's mentioned in that citation, Ann Madden, she is hopefully going to be one of our guests. Am I giving it away?
Maybe in the long future.
Maybe. Oh, she's so busy, huh?
she's busy.
Good.
All right. Okay. Let's talk about what's the difference between this and baker's yeast. So baker's yeast, which most people are using to make their dough. How does that differ from sourdough? So what's different between all of this that we've talked about so far? The important thing to realize is that, like I was explaining, this high diversity of species that is microscopic, it's hard to understand how much diversity there is without really spending a lot of time kind of getting into this microbiology. So kind of as an anecdote, I've added an analogy here between animals that we know so that we can try and understand how different these things are. So a baker's yeast, there's usually around 3 strains that are used in baking. And it's kind of like if you just had 3 different colored labs. So you've got your brown lab, your white lab, or your black lab, your white lab, and your brown lab. And you're really just operating on those 3 different dogs. It's like if everybody had just those 3 dogs and there were no other dogs on the planet. What you have in your sourdough, it may also be called Saccharomyces cerevisiae, but it might be a different strain. And what a strain is, it's in the same species, but it varies in what it eats and produces, the temperatures it grows at, and all kinds of other things. And that's akin to if you were to have now every other dog breed opened up, but one step further, every breed of wolf as well. So that's how much diversity is encapsulated just in the name Saccharomyces cerevisiae, in that the baker's yeast you might be using might be completely different from the Saccharomyces cerevisiae, even though it has the same name found in your sourdough. They could have very different properties that could even matter to a baker, such as, ones in your sourdough might be more stable across lots of different temperatures. And so you get a more consistent fermentation across different temperatures, for example. So it could be beneficial. And then on top of that, you now have tons of other species of yeast in your sourdough as well. So not only are there just the explosion of the different strains, like wolves and other dog breeds, you have foxes and sea otters and badgers and a walrus and a bear and a panda and all kinds of other species that are found within kind of a similar group that can also be found in your sourdough that do totally different things. But if you were to look at them under the microscope, they might look pretty similar. It might look like that budding cell. But importantly, they could be performing very different functions. And then it's really hard to make an analogy between how different a dog may be from your bacteria, because there's no other way to describe it other than what we know here on Earth, which would be the difference between a dog and a bacteria. But it's kind of like a totally different branch of life that has totally different functions and metabolisms, like an alien. So I've got Chewbacca there. Scientists, forgive me. But the point being that these microbes, these bacteria function totally. They live in thermal vents. They can live off sulfate only. They can live in radioactive vaults. They can live in space. They can live all kinds of places that eukaryotes and animals can't. So it's hard to describe how much diversity there is in there, but hopefully this gives you some intuition. Good, Noel?
I mean, I think it's great. No, I think this is great because when we started to talk about this literally months ago, I didn't really understand what you were getting at. And so I feel like these kind of simple graphics where you use, groups of animals, photos like this really helps me begin to understand the kinds of diversity you're talking about, even though this is really just a very kind of crude animation, right?
Yes, yeah. Scientists might be slightly mad at me.
We're not mad at you. We're happy because you're bringing us into your world, and you're doing it in a way that I find approachable. So thank you.
Awesome. All right. We're going to spend only a little bit of time on this slide. We could talk to the people who made this graphic and spend an hour just talking about this because it's awesome. But this is a study that I pulled, and I have a link to the study here, and we could give you the link as well, where they collected 500 sourdoughs, mostly from the United States, but also from Europe. And what we're looking at here is 2 bar plots stacked on top of each other. The top one is the lactic acid bacteria and the acetic acid bacteria. So those represent the bulk of your bacteria that make lactic acid and bacteria that make acetic acid, which we didn't talk about, but we can later if people are interested. And then on the bottom, we have the yeast. And when they fill the whole bar here, it's only that one there. So it's 100%. So you can have, 50% of one and 50% of another microbe. And they're colored down here. So for example, if we look at this sourdough starter, we have mostly this pink here. And so that one is mostly Lactobacillus sanfrancensis. It's hard to say. And then we have this gold color, which you guessed it, Saccharomyces cerevisiae, our old friend. But the point being here is that there's all these different colors encapsulated in these bar plots. And this is just to demonstrate that all of that diversity that I talked about can be found across all of these sourdoughs. And so your sourdough might not behave exactly like another person's sourdough because it's got a different set of life in it. You may have a gray wolf and Chewbacca, and another person may have, a polar bear and what's another good alien there? Spock. So if you're comparing one starter to another, it can be difficult, even though we can try and break them down into these kind of metabolisms, and the set of them are pretty well conserved. You can see there's not that many different ones, but they do differ from sourdough to sourdough. So this is kind of just a 10,000-foot view, and we could talk about this for days if we wanted to. Questions? No.
I would love to talk about this for days. So let me try to understand what I'm looking at, because it's a little bit of a blur. So is the idea like if you had like 500 samples, this is kind of like a representation of what that would look like, or 10,000? I don't know what the number would be. This is kind of
this is the way you can think about it. If you just look at this bottom part, this is a good representation of most likely the bacteria and yeast that you're going to find in your sourdough. If we have 500, we can make a good guess that in your sourdough, it's probably one of these yeasts and one of these bacteria. Most likely, you'll have a strain of Saccharomyces cerevisiae and 2 to 3 lactic acid bacteria, and maybe an acetic acid bacteria.
So LAB is lactic acid bacteria, and AAB is acetic acid bacteria. Got it.
Yes. Yeah.
All right. Well, I think we're going to do a Zoom just about this.
Yes, we could.
Good. Thank you for that.
Okay. So we're getting close to the end of the science, and we're going to get to the practical part. But the last thing I wanted to highlight is where do these microbes come from? So this comes from a great paper, again, linked down here. We'll send it. But what they asked was what microbes contribute to a sourdough. So when you first make your sourdough, you have an island. You can imagine it like an island floating in the ocean. And they want to understand which animals will come colonize that island and start to thrive, and kind of why would be the ultimate guess. But let's just understand where they're coming from first. So they sampled people's hands, they sampled the flour they used, and they sampled their built environment, meaning their house. So they sampled their walls and their kitchen floor and all of those types of things. And what they were looking for is which percentage of microbes come from where. And so the first place they come from is your hands. So around 26% of the microbes found in your sourdough come from your hands, and vice versa. So this is the coolest part, is they then sample the baker's hands again later after they started making their dough, and your hands start to become more like the bread that you're making. So bakers who spend a lot of time touching their dough, your hands might look slightly different from a non-baker's hands and more similar to your sourdough over time. So you're feeding your microbes to your sourdough, and then your microbes, or your own microbes in your sourdough, are then recolonizing your own hands in greater numbers. The next place it came from was the flour. So around 60% came from the flour. So a lot of the microbes come from the flour. And then around 14% came from somewhere. We don't know. The house, the air, a dust, who knows? Maybe a fly landed. Who, we'll find out. Maybe in the future they'll do a bigger study. And that's another thing to point out here, is that a lot of these studies are N of 1, so under a researched field, and they need more funding, and they can do more with that and generate more studies. Usually in science, we like to repeat things multiple times before we solidify them. That's why there's this little squiggly mark here that means about. So that number is subject to change until they do more studies. But I think the bigger question, right, is this 60% of microbes. Where do these come from, right? Like flour's a lot of things. What contributes to that flour? Well, a lot of it comes from insects, actually. They move from plant to plant and leave their microbes behind on the outside of the plant. And when it gets ground up in the mill, those microbes get ground up with it. They're too small to get smushed, so they end up intact inside your flour. And you probably have experienced this before, where you go and leave your flour for a long time at room temperature, and then it starts to sprout flies. That's because they're working their way in there, and they've survived through the milling process. It comes from the plant itself, not just the insects that land on it, but the plants that are the microbes that are indigenous to the plant and the soil that it was rooted in. And then it also comes from your house/bakery. Your dough is touching the surfaces, your spoons, all kinds of places, and there's microbes that live inside your house and can colonize your flour. No,
it is crazy to me. This has been like bending my mind for weeks since you first showed it to me. When you say that the flour is 60% approximately.
Yes, approximately. Important.
We're talking about the flour that this started with, or the flour that you're using to feed the sourdough? Which are we talking about?
Yeah, that's an important distinction. It's the flour that you initially made your starter with. Once you make your starter, this is still something that needs to be studied more deeply. But once you make your starter and it's kind of stabilized, you passed it for 10 days in the same flour or whatever, at that point, the community will start to solidify and it's harder to disrupt. Meaning, you have a stable ecosystem there, and it's really hard to disrupt that unless there's an outside force. We'll talk about some practical reasons why that might happen in a second.
That's crazy. So if one person shared the same sourdough starter with 10 people, right? Each person's hands would be responsible for about 26% of where that starter ends up. But the flour they're feeding it, right, is not as responsible as the flour it was started with, right, within that 60%. Correct. And then 14% we still don't know, and we're not saying.
Right. Yeah. Approximately, with one study. Maybe in the future we'll get another one.
I mean, it tells me that there's a lot we don't know, and when people boast about their starter being whatever they're boasting about, they're really just boasting maybe about a very small percentage of what's actually in their starter, right? Like how old it is, right?
Yeah.
I don't see age here. Where's age?
We can maybe cover age in another one, but yes, there was a study that kind of looked at age and didn't find a whole lot. Maybe 1 or 2 microbes slightly changed, but
that's very interesting to me, because that's about the biggest brag I've heard, right? My sourdough's 1 million. Oh, mine's 2 million. Oh, it's funny, mine's 1 billion.
Yeah. Most likely doesn't matter that much. But again, N of 1, we need more studies like this, and people are generating them.
Good. I'm glad to hear. I mean, this study is only from 3 years ago, right? 2020.
Correct. Yeah. I have some resources at the end of people who are working on this in academia and generating these data sets that are so rich.
These are awesome. What about geography? That's another thing people brag about. Like, my starter's from Los Angeles. Oh, mine's from Italy. Rome. Really?
Yeah.
How come location isn't on here?
Again, global geography doesn't seem to play a huge role. Again, N of 1 or N of 2, there's a few studies that have shown this. But what could play a very large role is your local environment, right? Your house and your bakery. So a lambic brewery, for example, has been shown to give a lot of acetic acid bacteria to the sourdough starter, which changes the flavor, makes it a little bit more vinegary, depending on how you use it. It could be more green apple kind of flavor.
Wow. One of the chefs that I've been working with for many years, he worked at Bestia in Los Angeles after I did. He took one of Ori's sourdough starters to Florida with him for a project. And one of the things that was incredible, and I don't know why this happened, was that suddenly that same starter that we used to feed maybe 15 or 20% as a percentage of flour weight, you only needed to feed a couple of percentage points because it became so active. Right. Yeah. And we don't know we thought it was just based on the location, but based on just this one study and these very, very kind of crude estimations, there's a lot more going on than just moving from Los Angeles to Miami, right?
Right. Yeah. We'll cover in a second some of the things that could happen. But yes, a lot can happen that you would never know happen.
All right.
It'd be very hard to tell.
So is this where we're going from the science to the practical? Where are we at right now?
Yes. And we kind of just captured this, is that, okay, we're kind of driven ourselves crazy. We've got Charlie Day there trying to figure out a conspiracy. We've covered a lot of ground. Hopefully everybody's ready to do some practical stuff. Can we translate some science past the paywall and hopefully give some useful information? But again, number one rule with this is that making universal rules about how to manage your sourdough, and I'm sure bakers understand this better than anyone, it's difficult because we don't know which species are in your starter. And that could change the way that you need to manage it. And also, a lot of this research, only has one or two representations of these results. And so, someone could do a new experiment and prove it wrong. That's what happens with science. Things get updated all the time.
Love it.
All right, let's jump in.
Please.
Okay, so common observation number 1, temperature and hydration. So I labeled it common observation for the reason I just described, is that things can change. But generally speaking, if you have a lower hydration and lower temperature fermentation in your starter, so you keep it at a lower hydration or lower temperature, it will favor the yeast and the heterofermentative bacteria, right? So these are the microbes that will contribute the most CO2. You'll get a less lactic-y, less dairy notes in your sourdough, right? As opposed to if you keep it at a high hydration, right? Like at 100% hydration and higher temperatures, you're going to favor mostly these homofermentative bacteria, which, remember, we talked about only produce lactic acid. And so you're going to get less leavening power, so less CO2, less bubbles, and you'll get a more kind of yogurty flavor to your sourdough. Furthermore, if you keep your sourdough at above 98 degrees Fahrenheit or 36 degrees C for a long period of time, which can happen, right? You leave it at home, you didn't turn on the air conditioning, it's 110 degrees day, you live in Phoenix, you can completely lose the yeast in your sourdough forever. The yeast can just drop out, and it will either just continue on with just the heterofermentative microbes will make our heterofermentative bacteria will make your CO2, or something in your house will replace it, and your sourdough may change for the rest of time. I don't think anyone, unless you were isolating your microbes and storing them, and you want to reconstitute your sourdough, which I think is unlikely, you've kind of lost that yeast forever. But again, making rules in biology is hard. You can break this trend completely. If you had Lactobacillus plantarum and Fructi-Lactobacillus sanfranciscens, those could dominate a stiff starter and completely change this on a to be the opposite of kind of what we've just laid out, right? So if you tell your friend, you know, this is what I see with mine, and they say, well, I see the opposite, you guys might have different bacteria in your starters. All right.
That's one of my favorite takeaways, because I got to say that I've been doing this professionally for about 15 years, and up until now, it's confounded me where I'll be speaking with another baker and we'll have opposing views of the same thing, only with regard to sourdough. Things like hydration and temperature, and we're all on the same page, but we're getting literally opposite results. And it's been driving me crazy, right? And on another level, my students and my clients are always saying, Noel, here's what I need. I need a ratio, right? You got your commercial yeast over here, instant dry, active dry, fresh. You've got your sourdough starter over there. Like, what's the ratio? If I want to go from instant dry to sourdough, if I want to go from fresh to sourdough, if I want to go from sourdough to active dry, how do I do it? And I always just say, well, it depends. How fresh is your fresh yeast, right? How active is your active dry? How active is your sourdough? And there's so many moving parts. Now I see where I was literally not even scratching the surface when I gave them these answers, right? Because they don't do any of what you just described justice. So I could actually spend a lot of time talking to you about this, but we are on a tight schedule. So let's go to the next point.
Yeah, let's finish our practical and then we can keep going. Okay, so salt. This is a common question, I think, is, you know, what's salt concentration going to do to my sourdough? So a lot of people just don't put salt in their sourdough at all, from what I understand. I'm only an amateur baker, but from the science side, what can happen is if your salt concentration is above 0.7%, there's kind of an inflection point, at least in this sourdough that these people study. Again, things can change. And that's a percentage as total dough weight, not a baker's math percentage. So make sure you take note of that. But what can happen is as you increase that percentage, you can pretty much totally lose or select for a certain set of bacteria relative to yeast. So what will happen is that as you increase this salt, so kind of around 0.7, the bacteria will increase a little bit, and then they just start to completely drop off. So at this axis, we're looking at the total amount of bacterial cells in the sourdough across temperature and then across the salt concentration. So NaCl is the scientific term for salt. And you can see as we get to like a little over 3% of salt, we've almost completely lost our bacteria at most temperatures, very small. And our yeast stays about the same. So what happens is you have a much more dominant yeast community over your bacteria. So you get a very yeasty dough, almost like you're just probably more like similar to if you were to be using just baker's yeast or something like that. You're gonna dominate your yeast out.
This is fascinating to me, because I've always understood salt as something that helps to concentrate or highlight flavor, but it turns out it's a lot more than that, huh?
Yeah, at least within your sourdough starter.
Yeah. And it also brings up another interesting point, which is that most people don't put salt in their sourdough, but our friend Daniele Uditi from Pizzana, he does. Yeah. And makes me wonder, I mean, his pizza is some of my favorite in the world, right? And it just makes me wonder, maybe he's onto something. He's from, I think he's from Caserta, which is a village near or just outside of Naples, and he's from a baking family. So, you know, he's the only person I know that has a salted starter, and it makes me wonder out loud, like, what does that guy know intuitively that you know scientifically, right?
Yeah, he could be selecting for a select group, or if he has a really small amount, he could actually be increasing his bacteria a little bit.
Awesome. Beautiful. I love this.
All right, last one, and then we'll answer a few questions and hopefully be able to answer some more. Feeding your starter. So the larger the percent of starter you use, it favors yeast growth. I think most bakers probably know this one. While a small inclusion will favor the bacterial growth. So somewhere between 30 to 50% as a total dough weight will favor the yeast growth. You get a really yeasty, again, similar to if you have super high salt, you'll get a very yeasty, highly fermented dough. And then if you feed somewhere between 2 to 10, you'll be able to favor your bacterial growth. So you'll get a little bit more flavor, a little bit more sourness from that starter that you're using. So not only does it change the time and the refresh rate, but it also can change which microbes are dominant in your starter.
So this just to make sure we're all on the same page here. This is not, you're not talking about 30% of the total dough weight, 30 to 50% of your dough, of your final dough. You're talking about 30 to 50% of your dough weight in the sourdough starter feed.
Your total final dough weight. So a percentage of everything.
Got it. Okay, good.
Yeah. So not bakers. Got it. The scientists don't use the bakers for some reason.
That's all right.
People to read.
I mean, it's all baker's math, whether it's you're talking about your percentage of the flour weight or the percentage of the final dough. It's just a different way of thinking of percentages because you have percentages that can go above 100%, right?
Yep, exactly.
There you go.
All right. So very quickly, there's a lot of things that you can't control. So you can't control which microbes are colonizing your starter to start with, or if one of your yeast drops out, or all types of things. You can't control what's in your house, the microbes in your house yet. It doesn't matter how much you clean, they're still there, I promise you. So you can't control what might jump in if your yeast falls out because it's too hot. You can't control what's in your house and might jump into that community if it becomes unstable. And you can't control what we call the biotic factors, which is the community in there, the relationships between the microbes. You have no control over that, really. You can kind of control their abundances with these parameters that are abiotic or outside, factors like temperature, but you can't really control the direct relationships between these microbes. And that's just the tip of the iceberg and kind of things that you can't control with temperature and time and things like that. There's it's amazing that you can control as much as you can and that people have learned to control it as well as they have. Probably the best example of that I know of is human food fermentation, of the relationship between humans and microbes outside of what's living in your gut, maybe. But you don't have, outside of diet, you don't have control over that either.
So I'm trying to look at that image of the iceberg, where obviously you have a very small amount above the surface of the water and a very large amount below. So help me with the symbolism here. So what's below the water surface is stuff that we can't see.
Yes. Yeah.
Don't understand. What else?
Yeah, hasn't been studied yet. No one's thought to even ask the right question, right? Most of science and probably baking too is, did you ask the right question when you did your experiment? If you ask a bad question, you're just wasting money. You're burning money piles at that point, right? You need to ask the right question first, and so it takes the right people to come along and come up with creative questions and the right questions to ask. And it takes bakers and scientists talking to each other.
I love that. That's why we're doing this. I gotta say, one of the things that I hate, I'm sorry to use that word, Justin, you can correct me. One of the things that makes me uncomfortable is when people repeat this mantra, right? That, that's because baking is a science. I'm like, I don't think baking is a science. I'm not a scientist. I'm a baker. I think of myself as a crafts person that has really no knowledge of science. My father was a chemical engineer, so we had fun with exploding stuff in test tubes when I was a kid, right? I took basic biology in high school, but I'm not a scientist. I barely understand what we're talking about right here. What I do understand is super basic relationship of numbers: addition, subtraction, multiplication, and division, right? A little bit of ratios, maybe some fractions here and there, the ways that temperature and time relate to each other. But this is not science, right? What you're talking about is science, right?
Yes. I'd say, if you take really good notes and you're very meticulous about things, you can You're getting close to science. We're good at note taking.
You're definitely good at note taking. I mean, this is just me, my personal opinion. I feel like baking, as I understand it, is a craft. And depending on what you bring to it, whether that be science or maybe an artistic eye, right? Maybe an unbelievable palate because you're a chef, right? I don't know. But everybody brings something different. And the science right now is a very small element of what artisan baking of pizza and bread is. And I would just like it to be a more integral part and have all of us have a better foundation, right? Math, science, art, music, it all kind of comes together in this beautiful craft that I think of as artisan baking.
It's a great way to put it. It's beautiful.
I try. Thank you.
Okay. Really quick, Justin, we got time to answer some questions that were submitted.
Yeah, you want to start with the ones that you have here, and then we've got a few questions from the chit chat here live.
Yes. Okay. So the first one was, can you buy other types of yeast shelf stable? So in a packet like you get baker's yeast. Generally speaking, no. So most of the microbes that you find in your sourdough, you can't find nicely preserved in a packet for you. Even the freeze-dried microbes, we don't truly know what survives in those pastes. So, you might not be getting exactly what's in there. There are some species and strains that, are used in other areas like brewing and beer and wine, which could use in bread. So like Saccharomyces bayanus, you can buy it on Amazon. It's shelf-stable format. There's some studies, I cited one down here, that's citation number 1, where they used it to ferment bread. And, if you do it carefully, you might be able to make bread with it. The second one is, if you purchase a sourdough, will local yeast eventually take over? So this is a difficult question to answer because nobody's really done it. So the true answer is, I don't know. But if you force me to answer, my best guess based off of other microbial ecosystems is that the mature starter will be relatively hard to influence. The community will be pretty strong. They've kind of formed from all of the microbes when you made your starter to exist together because they have a good relationship. But if you create a factor such as storing your starter at 98 degrees Fahrenheit or above for a long period of time, your yeast could fall out, and then your home microbe, like I said, could take over, might replace your yeast, or there just might not be a replacement for that, and the bacteria might just exist without the yeast for a while. And then the last one, also a difficult question, but what are the effects on each if commercial yeast is blended with sourdough and dough? So it depends somewhat on the species in your starter, right? Again, going back to, we don't really know what's in your starter, so it's hard to answer. So some species with more acid will produce more acid with baker's yeast. So for example, on the opposite side, Lactobacillus brevis, which certain strains of that require maltose to grow, just like Saccharomyces cerevisiae. You'll probably just get a domination of Saccharomyces cerevisiae. In other cases, your bacteria in your sourdough might not be Lactobacillus brevis, might be something else. And that one might grow well with Saccharomyces cerevisiae because it eats a different sugar than it. And so it won't be competing for nutrients. It might even be beneficial. So it's impossible to say unless you know what's in your sourdough. You can do some experiments at home, maybe by just adding it and seeing how sour it is after you add the Saccharomyces. If you added it enough, it will just take over. Okay, so happy to answer more questions. There's a whole set of other information here. So a whole set of books on just microbiome in general, microbiology, some basic machinery of life, like you were asking about sequencing and how that works. This is a great, beautiful, illustrative book about how that works. This book, I Contain Multitudes, is an amazing book about microbiome in your gut, all the microbes that live with you. Never Home Alone is about the microbes in your house. And then if you want a textbook, Biology of Microorganisms is a classic. And then there's handbooks for sourdough specifically that are super scientific and useful. And then a whole set of free and easy to access information on sourdough. I highly recommend, especially Rob Dunn's lab, who generated a lot of this research, has a great kind of informative and with pictures and everything on all of this research.
This is great. Thank you.
Good.
Yeah.
All right. I'm going to stop sharing.
Beautiful. This is awesome. So, guys,
hopefully,
we're going to
be... That's a lot.
No, okay. What were you going to say, Cameron?
No. Sorry. Go ahead.
That was fantastic, guys. I just wanted to say that was absolutely stunning. Can I bring Dom on? Because Dom had a question. Is that all right? Yes. So, Dom, I'm going to bring you on right now, if you don't mind. If you want to go ahead and unmute, buddy, thank you very much.
All right. Sorry about that. How's it going?
Going great. You seem to be popping up every week. You're at the sourdough sessions. You're at the office hours. Thank you for coming back. Thanks for your support.
No, I've learned more than I could have ever imagined over like the past 3 weeks. So I kept coming back, and now this kind of seems to have answered like an issue that I dealt with last week. Basically, my wife baked my sourdough starter on me an hour before I had to use it. So I was scrambling last minute, and I picked some up from Raza in Jersey City, and I brought that home. I fed it. It rose like my starter normally would have. I mixed my dough like I usually did. I did 4 sets of stretch and folds over the course of 2 hours. I let it rise again for about an hour until it hit about 20%. And then I put it in the fridge for 24 hours. Normally, I take it out, then I ball it, and then I put it back into the fridge. I didn't have enough time to do that, so I balled it and I left it out to just do its final proofing, and it just would not proof. It took forever. Like normally, I would say when I take it out of the fridge, like within 3 hours of it being at room temperature, like it's ready to roll. And this took probably like 18 hours. And like in the beginning, I was freaking out because I was doing Detroit style pizzas. The dough, it just kept expanding in the pan. And I was like, I'm just going to do a test bake, see what happens. And like, it was a pancake. And it was pretty rough. So I feel like the bit on the whole time and temperature, like that might not necessarily be the case. Might have been what happened, because I was just like, I don't know what could have happened. It just wouldn't rise. It was strange.
Yeah. So for those of you, we're going to be posting the office hours from last week in a couple of days. So for those of you who weren't there, Dominic is a baker, and he has a little micro business going on with a pop-up. And like he said, his wife accidentally baked his starter. So in the office hours, we were discussing backup plans, right? A straight yeast. We talked about some ideas for poolish. I emailed you 3 different poolish recipes depending on the time that you had, and so. We also talked about maybe using a combination of sourdough and commercial yeast. And for me personally, in hindsight, I'm not going to say I was right or I was wrong, but anytime that you hand off your starter to somebody else, anything can happen. And I say that because everything that could possibly go wrong or right in my mind has happened to me in the last 15 years. Maybe half of it I think I can explain, but I would say based on what Cameron's been saying, I can probably explain 2% of it. Right. And hopefully, go ahead.
Oh, no, I was going to say, fortunately, I know I had emailed you back and I said that I ditched the poolish, and I did. But I also, halfway through the day on like Wednesday, I was like, you know what, let me just do another one just in case. And I did have another one that I was able to fall back on at like 3 a.m. to get the dough ready to go. But yeah, it was a wild ride last week for sure.
Wild is the word. Thank you for sharing.
Yeah. Anytime.
Beautiful. Thank you. All right, Cameron, we're going to start winding this down. Why don't you say some final words if you have any, and then I will say my thank yous, and then I will pass the torch to Justin. What do you say?
Yeah, thank you everyone for attending. Hopefully everybody learned something useful, and I'm excited to see everybody in 2 weeks. Like everybody else, I'm excited to hear from Stephanie. I think I'll learn a lot there.
Beautiful. Thank you. Let me thank James, who's been working tirelessly in the background with Cameron, putting all of these references into the chat and answering some questions. So for those of you who are not catching us live and watching us on YouTube, or maybe listening on whatever app you might, this might end up being a podcast, who knows? We will try to include as many of the references that we cited in the description below. So please have a look if you're interested in finding out more about any of the things we've talked about. Also, this is a podcast that goes every other week. I use the word podcast. It's a live Zoom for right now, free, and we try to keep it to about an hour with maybe 10 minutes of Q&A at the most. So thank you everybody for participating. Of course, Justin, thank you for keeping things moving and right on target. So I'm looking forward to doing this again in 2 weeks. Justin, I pass it to you.
Thank you, buddy. I just want to say thanks to Cam and thanks to Jimmy for putting this on. This was really fantastic, really enlightening. A lot of information. I know that we're going to have to digest it like a nice piece of sourdough. You know, that's. There was one question Dennis was asking about any recommendations on starters that maybe he could find in the SoCal area. Noel, any thoughts? Talk to our buddy. You know, we got some friends who are doing some sourdough stuff.
I mean, you could always go to Joey at Folks in Orange County. He has a starter that he's happy to give you. And you know, if you want to just make it from scratch, there's lots of great videos on the internet. We have a friend named Sourdough Duffy. I met him when I was up in Toronto doing a consulting project up there. He is a wealth of information. So I believe he's what is he? He's Sourdough Duffy on Instagram. I think you can find him there.
So yeah. And Kate is saying she's happy to share her starter. She's down in San Pedro. So thank you, Katie. I know you've been coming to a couple of these classes. Guys, we're going to wrap it up here. Thank you all so very much for coming to hang out with us. We'll see everybody next Monday at office hours, 5 p.m. sharp. You know the dealio. We do not get into the politics of pepperoni and pineapple. We say forever and ever, pizza always, and always pizza. Pieces and slices, everybody. With love. See you in a week.
Thank you, everybody. See you in 2 weeks.
2 weeks. Next Monday. Who knows?
Beautiful. Thank
Bye,
you,
guys.
Everybody.